All right, kia ora. Here's our second task for the week 5 of remote learning. So today, what I want you guys to do is define the new types of alleles. They are obviously much more complicated than just dominant and recessive, and that's what we're going to look at for today's lesson. And I also want you to explore then how does that influence their inheritance and, the expression of those phenotypes. Cool. So from the success criteria today, you should be able to describe and explain a lot of new vocabulary that refers to things such as incomplete dominance, codominance, multiple alleles, and lethal alleles. Really important that you understand the difference because they're probably going to ask you questions on these and ask you to do test crosses and pedigrees with this information. So let's look at what is a little bit more accurate to what is the story with genetics. So, generally, the first thing to think about is we have lots of different phenotypes, but those phenotypes are not often a simple, here are the two alleles, one allele is dominant, one allele is recessive. On top of that, multiple genes can control one phenotype, and so you have different layers to then that result then in the final trait. So the first factor we need to think about is different types of interactions alleles can have, such as incomplete, codominance, lethal, and sex-linked. The second thing that we need to factor in is multiple genes controlling that phenotype. Cool. So the first one I want to show you guys is incomplete dominance. Normally I would give you guys this as a describe and explain, but I am mindful that we're in remote learning, so it's probably just going to be me explaining. What you should hopefully notice when you're looking at this, and just as a reminder, we have the P generation, which is the parent generation. We have the F1 generation, which is the offspring, and then the F2, which is the second generation from that. So what we have then in our alleles now, instead of just using the typical capital letter, lowercase letter, we're changing it up. We have C to represent color of the flower, but then we have a lowercase r and a lowercase w, and that's referring to red and white. And I'll explain in the next slide why those are lowercase letters, but I don't think they're going to be too picky and if you draw it as a lowercase or capital. What you should notice is that when I cross a red flower with a white flower, everybody in that next generation is pink. So we see a blending of the two traits. Then if I take two flowers from that generation, that F1 generation, I notice that it then expands back out. Most of my flowers are still pink, but I do have some red and I do have some white. So I start to see the parent generation's phenotype again. And the reason why is because of something called incomplete dominance. So this ties back to why we're using these lowercase letters. It means that neither of these alleles are the dominant one, and instead what you're going to get is a blending of the two. So basically the heterozygous outcome looks, as I said, a blend. So red and white makes pink, and that's why we have that. Cool. I don't know if you guys need a Punnett square for that. I'm sure there's questions in the homework and stuff like that that will help you practice it, but basically when you do the Punnett square for this one, 2/3 are heterozygous, one is the homozygous for red, one's homozygous for white. It's the same Punnett squares that you're used to seeing. All right, the next type of example is codominance. Again, you're supposed to describe and then explain, but I'm probably more so going to explain. So again, the P1 generation, we have healthy cells that are normal oval shape, and then we have deformed cells. When those two parents breed together, we have everyone with a mixture of healthy and normal, and then when two people with healthy and normal breed together, you then have that full range of options, either healthy cells, mixed cells, or deformed cells. This example here that we're actually thinking about is sickle cell anemia. So let me first explain codominance and then I'll talk about sickle cell. So codominance in this case both of the traits are dominant, and so they both get expressed, and so we get a mixture of both traits. So the difference we have to remember between incomplete and codominance is that incomplete is a blend, codominance is both of them there. And we represent that by capital letters because they're both dominant. So R for regular blood cells, S for sickle cell. And like I said, we did talk about sickle cell in that video, and we noticed there was that girl who was really, really sick because her red blood cells weren't carrying enough oxygen, and because of the funny shape, they were getting trapped. And that's that right here. Really, really potentially bad, obviously. So for her to be able to get that, both her parents needed to be carriers. They had that mixture of normal and sickle cell. They don't feel the same effects of sickle cell anemia because they still have some normal red blood cells available. And what's really interesting, and we'll see this in the population genetics, is what is the evolutionary advantage to having deformed blood cells? Sickle cell anemia is very high in African populations, and the reason why is it ties into malaria. So malaria is a blood disease that's passed by mosquitoes, and malaria can infect healthy cells very easily, and they basically hide inside them and burst them open, and you die because you're not transporting enough oxygen around the body. The malaria parasite cannot infect the deformed red blood cells that well. And so it basically provides a layer of protection. So if you have sickle cell anemia, you're not going to get malaria. But the problem is then you have sickle cell anemia, which has its own problems. So what's interesting about this is that this trait evolved because individuals with that mixture of healthy and deformed cells had a layer of protection against malaria. So these people here would die from malaria. These people here would die from sickle cell anemia. These people would survive for both traits, or both diseases. So that's where it came out from. And we'll see some, like I said, population genetics showing that. Cool. The next one here then is lethal alleles. And I guess I was planning to fill this one in. Or having you guys fill it in. So a lethal allele, the hint whenever you see a lethal allele is that the ratios don't make sense to what you predict typically in a Punnett square. So the lethal allele that we're looking at today is with cats, and these cats have no tails. I should actually find a picture. So let me just pause so you know what I'm looking at. Cool. All right. Any opportunity to put a cat photo in, I'm going to take it. All right. So this is just an image to show you what these cats with no tails look like. It basically looks like there's a little stub there. And there's just no tail there. So obviously most of us see cats with tails, and this stub isn't the result of any sort of accident. These cats are born this way. This trait is actually heterozygous, so it's T, little t. And if we breed these two together, you would think with the traits and how they come out, let me just center that. So I should have T and T, one of them red, one of them blue, just to help. I should have T and T, so one big T, one little t in that box. One T and a little t. And a TT. Little t. It doesn't actually matter to have the colors in there. I just find the colors help. All right. So now I filled in this Punnett square. And what we are noticing is that we have homozygous dominant, we have two heterozygous, and we have homozygous recessive. In our previous interactions with heterozygous traits that were just dominant and recessive, these three would result in the dominant phenotype, and this would be the only one resulting in the recessive phenotype. So you would expect the typical 3-to-1 ratio, no tail to tail. However, when you look at the population of this offspring, you notice that there's actually a 2-to-1 ratio. So we're missing a fourth of our cats. And the reason why we're missing a fourth of our cats is not to do with the fact the sample size is too small. It's the fact that there's a lethal allele that's killing the offspring. So this homozygous dominant trait, when you get two of these big T's, instead of that producing a cat with no tail, it it just kills the cat instead. And it kills it in utero, so it's not going to be born or anything. And the reason why is that this dominant allele here is important for the spine development of the cat. And so when you're heterozygous, you only have a partial error in that spinal creations, and you're just missing your tail. If you have both capital T, then the whole spine then doesn't develop properly, and obviously the cat then can't survive. The recessive trait is to have the tail, homozygous recessive. And again, this is trying to prove my point that I was talking about earlier. Don't refer to things as being more common or stronger or weaker, because most cats do have tails. So this recessive allele is very common in the cat population, even though it is recessive. So I shouldn't even say even though it is recessive. Don't confuse that recessive means less likely. It doesn't. So that's a little kitty cat story. So if you notice any questions stating that the ratio isn't as expected, there's probably some lethal allele there that's killing off the offspring. All right. Next one is a little quiz time. Now, it's not obviously a real quiz. It's just to see if you can see the numbers. So it's basically just a color blindness test. So see if you can see that number. Can you see that number? Can you see this number? Can you see that number? Can you see this number? And there's another one. Can you see that? Cool. So this is, like I said, a color blindness test, and it's just used to test for red-green colorblind. And the answers you should have gotten were 16, 2, 5, 42, 7, and 29. The reason why I wanted... Oh, sorry. And then I have a question here. I remember now. So color blindness is much more common in men than it is in women. And we need to think about, why is that the case? The reason why has to do with where that gene is located. So the color blindness gene is tied to the X chromosome. This is really important then for inheritance. So since males only have one X chromosome, they only need to inherit one defective allele to be colorblind. The color blindness is a recessive trait, and because males have to get the Y chromosome from dad, because dad's the only one that can provide Y, X has to come from mom. So mom's the only one that can pass on the defective chromosome. Females, on the other hand, need to have both X chromosomes to have that recessive allele. So they would need to have a colorblind father and a mother that was also colorblind, or a mother that was a carrier, so that she would receive both recessive alleles and be colorblind. So it's much more likely you'll find a male that's colorblind than a female that's colorblind. And then here is like our Punnett square to show it. Again, like I said, the Punnett squares are getting a little bit more complicated. But what we can see is the capital R means that, you're regular seeing, you're not colorblind, and a little r means that you are colorblind, or that's the colorblind allele. Now the female up here, she is a carrier. She can see color just fine, but she does carry the defective recessive allele. The male, on the other hand, is not colorblind, so has the main big R allele. Now with the females, because the dad is not colorblind, all of his daughters will also not be colorblind, because he's able to provide at least one working allele. With mom, she can give the working allele, or she might give the defective allele, and if she gives the defective allele, then this daughter will also be a carrier. We won't know if she's a carrier or if she is a carrier unless she has offspring, and if she has any male offspring. For the male, on the other hand, dad can only provide the Y chromosome, and so once again, mom's going to be the determiner of whether or not they have color blindness. If mom gives her working allele, then the son is not colorblind, and if mom gives her defective allele, then she is colorblind, or he is colorblind. So that's how it works. And this actually ties in to very kind of personal to me, my family, because colorblindness actually does run in my family. So my cousin Sean up here, he's colorblind, and my brother is colorblind as well. And I have a funny story with that. We were once sitting on the couch, and my mom was reading a magazine, and it had an article about colorblindness. And we were just reading through, and it basically had the same like dot number test. And my brother said to my mom, I can't see any of those numbers. And then my mom was like, Justin, stop being dramatic. And that was the end of it. The thing is, that was the end of it. And then when my brother turned 18, just before he was moving out to New Zealand, we wanted to get an eye test because his vision wasn't great and he needed to be able to see the lecture notes and things like that. And so as part of the eye checkup, and this is just to go to show how like crazy the American healthcare system is, like my brother never got an eye test done until he was 18. Anyway, he gets an eye test done. The standard thing they do with an eye test is they give you a colorblindness test, and he outright fails it. And it was absolutely hilarious. And then he turned to me and he said, see, I told you I was telling the truth. I am colorblind. Nothing we could do about it, but he just is colorblind. And he was actually really good at hiding it. He was very good at like learning and memorizing what red and green were supposed to look like. But anyway, he's colorblind, so that means my mom must be a carrier. And what's interesting about my mom being a carrier is that my Aunt Bonaya is also a carrier because her son's colorblind as well. So both of these women are carriers. And the only way they could be carriers is if my akong, my grandfather, who's not featured in this picture because he passed away, was also colorblind. So, and that was a rumor from what I remember being told, that they thought he was colorblind. And the science says that he must be colorblind, considering he has 2 grandsons that are colorblind. So that's really interesting. I also had a colorblind boyfriend when I was in uni, and that was actually really funny. I love to play the game Red or Green with him because he couldn't tell the difference. But it would have been interesting had we stayed together, and if we had children, I could be potentially a carrier from my mom because I don't know if I got a defective allele or if I got the working allele. I know I'm not colorblind because my dad's not colorblind, so I've gotten at least one working allele. But if we had kids, I potentially not only could have colorblind sons, I could actually have colorblind daughters. But my current partner is not colorblind, so that'll rule that one out. So depends on if I have sons and if my sons are colorblind, but I won't have any daughters that are colorblind. Cool. This also leads into some other examples that are really fascinating, which is hemophilia. So hemophilia is the inability to clot blood. So what happens is if you get a cut, you just keep bleeding and bleeding and bleeding and bleeding, and you could basically bleed out and die. It's because you don't make the platelets that are needed to clot to then stop the wound from bleeding. Now, this is a recessive trait, and once again, this is linked to the X chromosome. What's fascinating is that this is very common in the royal family because of all the inbreeding. And if we look at how the Punnett square plays out, this is going to affect males more than they affect females. So, for example, we have the queen, who is a carrier, and she is X capital H here, and X lowercase there, and X, X capital H, and X lowercase. So I'm just going to highlight her in red. So she can pass down either the working allele or the hemophilia allele. Then when we breed it against a male, let's say the male has hemophilia. So X, I gotta fix my trait. X, H. So that is hemophilia. Y and Y. Cool. All right, so hemophilia, really common in the royal family. If the, actually, I want to change that. Let's make this person, I made a mistake. That one should be a capital H. Okay. Oops. No. There we go. Okay. Cool. So let's say the king that I'm dealing with is doesn't have hemophilia. He's fine. He's lived to his old age. And then he gets married to probably his cousin, because this is the royal family, and she's a carrier. So what you would notice is that the females tend to be fine, because so long as they inherit one of the normal trait, they don't have hemophilia. However, again, that Y chromosome is really going to toss in for a number, because whenever you have that Y chromosome, you then don't have a backup allele. So whatever is on that X is expressed. So if the female's a carrier, she has a 50-50 chance of having a male with the hemophilia. And we can see that in the royal family, a lot of males died, and there was a lot of difficulty producing male heirs because of that hemophilia. They would bleed out, they would get an injury, and they would get internal bleeding and die. So that's just something to kind of look at. Cool. All right, and then the last of the sex-linked genes are the dominant ones. In this case, you only need to inherit one X chromosome with the allele. Males, it can only be from mom. Females, you can get it from mom or dad, and it's just inheriting one. It's very unlikely that you're going to have any real problematic dominant sex-linked alleles, because it obviously would affect quite greatly both genders, or both sexes, I should say. Cool. All right, next one to then think about are our multiple alleles. So these are traits that are controlled by more than one gene. Blood type is the example for this one. So blood types, there are 4 different possible types. There's A, B, and O. And there are 2 different things that are happening here. The first thing that we need to kind of keep in mind is that the A and B traits are dominant, and in fact, not only are they dominant, they are codominant. So if you have both A and B, you're going to express both traits. I, which is tied into the O blood type, is recessive. So let's fill in this Punnett square once again so we can see how it looks. So if I have that combination, so we see A dominant, and we have and B. Both of these are dominant, so this one is an example of codominance, so you're going to have the AB blood type in that case. In this case, you have the B allele, and you have the recessive O blood type. So in this case, this individual will have a B blood type. This one here has the A dominant allele from the mom and a recessive allele from the dad. So this one would be A blood type, and then this would be 2 recessives. So we'd have an 2 recessive, so this one will be O. So from these 2 individuals, we can actually get all 4 of the different blood types because they're both heterozygous for the A and then heterozygous for the B. Obviously, you can be homozygous dominant for the B and have 2 BBs, or you can be homozygous dominant for the A and have an AB. You can be AB dominant, or you can be II recessive. But the thing to remember there is that you have multiple alleles controlling this. Cool. Let's tie that into what that means. So basically what happens with these blood types is if you are blood type A, you express this protein on the outside of your red blood cells that say, I am an A. And then your blood will have antibodies to protect you, and it's called anti-B. So basically it has the antibodies to attack the B blood type. And this is just a quick way to detect foreigners, because the B blood type is obviously not what you should be having in your body. The B blood type has the antigens for A. So if you mix in some A into B blood, it's going to clot. So that's what those two blood types are. Now you get A if you have the A or homozygous dominant A, or you have homozygous dominant A blood type, or you have heterozygous A. So you have one of the recessive alleles. Same thing with B. If you get one of each, so let's say you get A and you get B, you have expressed both of the markers on your blood cells. Now, since you have both of those markers on the blood cells, you actually produce no antibodies in your plasma, which is a really good thing because you don't want your body to react to your own blood type. And then we have O. O is that recessive trait, and O has no antigens on the outside, and then their blood will be filled with both antibodies for A and B. So if A, B, or AB gets mixed into it, it's going to clot. Now, this is really important when we think about blood donations, because with the blood donations, certain things can mix with certain others. So O blood type is known as the universal donor because the O blood has no antibodies on it, so it can be given to anybody, and these antigens won't react to it because there's nothing on there to react. So that's why we call O the universal donor. We call AB the universal acceptor. So AB blood can accept any of the following, O, B, A, AB, because they have no antibodies in their blood, so they can just take whatever is given to them. If you're O, you obviously can only accept O because you have antibodies for A and B. If you're A, you can only accept A blood type or O. If you're B, you can only accept B or O blood type. In a situation where you are rushed and they need to get blood into you, they give you O because they know it's not going to react. If they give you the wrong one, like say for example B and your blood type A, you're going to clot and it's going to die. So it's really important to have these kind of understanding your blood typing. But worst case scenario, if they don't know your blood type and you're hemorrhaging a lot of blood, they'll just give you the O. What's also interesting when you look at the blood type is also the commonness of the different blood types. The good news is the vast majority of the population is O, and so they are able to donate to anyone. The next most common is the A. So right now, they're really desperate for A positive blood. They've been sending me messages to go and donate, which I'll do in a few weeks. But yeah, there is also that positive and negative thing. We won't discuss that, but it's just interesting to look at. But if you have O negative blood type, seriously make yourself a donor regularly because you can go to anybody. And if you're O as well, make sure you donate regularly. There's not really much of a use for the AB blood types because you can't really put AB into anyone other than AB. So I think that kind of shows somewhere AB can only go into AB. Cool. It is also a really cheap way to do a paternity test, these blood typing, because there's obviously certain combinations that are not possible offspring to parent. So for example, if both my parents have B blood type, there is no possibility to have A or AB because there's no A allele. You can have an offspring with O because both of the B blood types could be heterozygous, but you can't have one that's A or AB. So again, you can see the chances of what blood type you'll get depending on what each parent has, which I think is quite fascinating to look at. So if both parents are AB, there's no possibility of having the O because they both have the dominant one. So you're either going to be homozygous dominant for A, homozygous dominant for B, or you're going to be that heterozygous codominant. Anyway, I just think that's interesting to look at. And like I said, it's a very cheap paternity test. You don't have to get your DNA tested. You just got to check if the blood types make sense. Cool. Goodbye. That is today's lesson. So you should be able to know the new types of alleles and explore their inheritance. That includes codominance, incomplete dominance, multiple alleles, and lethal alleles. And I forgot to stick in sex-linked alleles, but that's also another one that you can do. So now it's time for you to move on to your Mahi choice. Like I said, it's on my Google site and all the resources are on Google Classroom. Do make sure you take a decent amount of time to practice this material because the last task for this week is a CPR quiz and you want to be well practiced and ready for that quiz.